Hi and Assalamualaikum Azminas and friends. Me, Noraida binti Zamri, will be presenting my FYP1 title, The Study of Biofilm Formations of Staphylococcus aureus Isolated from Human Skin. Let's go! Staphylococcus aureus is a gram-positive staphylococci that known to be a commensals and harmless towards the human body. The external barrier between the human body and the environment is the skin. Members of genera Staphylococcus, Corynebacterium, Streptococcus, and Propionibacterium compose nearly all the bacteria on the skins. Even so, upon breaking the skin barrier, they can colonize various parts of the body and cause infections. Infections can caused by the virulent factors of S. aureus. One of the major virulent factors is the ability to form biofilm. Staphylococcus aureus is a commensal bacteria on skin. It is known to be in the nasals. Commensal bacteria is typically safe toward humans. However, if they invade the human body through the break of skin barrier, it can cause infections. The infections are due to various virulent factors and the most significant factors of the ability to form biofilm. This study focus on the ability of isolated S. aureus to produce biofilms and the resistance towards the common antibiotics. This could be leading to the research questions. Can S. aureus from the commensals have the ability to form biofilms and be virulent? Are the commensals S. aureus able to resist killing by the antibiotics tested? The objectives of these studies are to isolate S. aureus from the commensals of healthy adult, to determine the ability of isolated S. aureus to form biofilms through using microtiter plates method, to determine the antibiotic profiles of the isolated S. aureus, and to confirm the identity of the selected S. aureus by 16S RNA gene sequencing method. This study is significant as S. aureus are known as commensals on the human skin, so they are safe. But there are the possibility that these safe bacteria also can cause biofilm formation, which is the virulence factor. Besides that, S. aureus may also have the possibility to be killed by common bacteria that use against the S. aureus infections. These studies can contribute to help better understandings on the virulent factors of commensals, S. aureus, and better ways to manage these bacteria. The external barrier between human body and environment is the skin. As shown in the picture, skin dominated by members of Staphylococcus and Corynebacterium will be focused on the nasals, as more than 60% of the S. aureus in the nose are found in vestibulum nasi. The infection caused by S. aureus are impetigo, which the red spots on skin, and when it heals is from a crusty yellow and honey colored. Next is cellulitis, a skin infection that causes redness, swelling on the skin. Next will be pneumonia, is the forms of acute respiratory infections that affects the lungs. And lastly is sinusitis, and inflammations of the tissue lining of the sinus. Examples of S. aureus virulent factors for proteins is protein A that capture IgG molecules and prevents phagocytosis. Next will be the coagulase. The coagulase is the S. aureus enzyme that is a clumping factor, act directly on the fibrinogen in the plasma, resulting in clumping. But most prominent factors is biofilm formation, that is a community of microorganisms that adheres to surface and form a protective extracellular matrix. S. aureus biofilms of formation happen when S. aureus proteins, including fibrinogen binding protein A and B, accumulation associated proteins, and surface binding proteins A enables S. aureus forms biofilms. Attachment, multiplications, maturations, and depressors are the 4 stages of the biofilm formation. The chart shows the history of antibiotics used as S. aureus been discovered. Penicillin first introduced to S. aureus and became resistant after a period of time. Methicillin then introduced to this bacteria and became resistant to the antibiotics, and since then, this bacteria called MRSA. First tests are the bacterial isolation that use a sterile swab passes through nostril and making a bacterial serial dilutions, then streak on the MSA plates. After that, it incubates for overnight. The result will shows a yellow colony. When it happens, it shows the SRS colony. The culture will be repeated on Na, which is natural alga, to obtain the pure culture. Next will be the 96 well biofilms assay. From bacteria grown in the broth, then the culture is diluted 1 to 100 in TSB is poured into the well. Then this will be go through, washed 3 times with sterile PBS at pH 7.2 in each well. Fixation with methanol for 20 minutes and left it air dry overnight. Lastly, stained with the crystal violets that used for gram staining for 50 minutes in the room temperature. Lastly, it can be measured at 570 nanometer using a microtiter plate reader. Antibiotic profile started by incubate 50 minutes in NB culture from the overnight NA culture. The bacterial growth then read the turbidity using spectrophotometer T100 and enriched for 0.08 to 0.13. The growth from NB streaks on the MHA. Antibiotic profiles using Kirby-Brewer disc diffusion method will be used. And then it will be incubated for overnight, and after that the result by measuring the hollow zones diameter. Lastly, application 16S RNA. These tables shows the guidelines to mix the chemicals in Eppendorf tube. Thermocycles then will be run as the table shows with different temperature that have a specific process. The forwards and reverse primers are used for the amplification, and the RNA sequencing can be obtained from the third party. That's all from me. Thank you for listening.